of the speakers.
Hi, everybody. I'm Tyler. Yeah, like Lucas said, if you can keep yourself muted as much as possible during the talks, we'll have a discussion session after the 3 talks. But you're welcome to ask questions either through the chat or just by raising your hand if it ever comes up. And then while Serge is pulling up his slides, I'm just going to give a quick introduction for each of the speakers, or for Serge to start. So Serge Dumoulin is a professor of perception, cognition, and neuroscience in the Department of Experimental Psychology at Utrecht, as well as experimental and applied psychology at the University of Amsterdam. He's a group leader He's the group leader of the Computational Cognitive Neuroscience and Neuroimaging section at the NIN, the Netherlands Institute for Neuroscience, as well as being the director of the Spinoza Center for Neuroimaging. He earned his PhD in neurology and neurosurgery at McGill University, and he did postdocs at McGill and Stanford. His work focuses on the intersection of perception, cognition, and neuroscience, and today he's going to talk to us about how population receptive fields properties change across the lamina of cortex.
Thank you very much for inviting me, and thank you very much for the nice introduction. Can you all hear me okay? Okay, perfect, thanks. So let's see. So it's a pleasure to be here, to see so many familiar faces and names, actually. So I thought I'd start a little bit with a more general introduction on my vision on sort of the laminar introduction. And I'll talk about different studies, but I'll sort of have to reiterate sort of the background of those studies as well. So the principal advantage of the 7T is, of course, that it gives us increased signal to noise. And I think that's the main advantage and the main goal for me. But you can trade off the increased signal to noise to go to higher spatial resolutions. And this is, of course, what we'll be using here and what we'll be talking about in experiments here. And if you talk about neuroscience, there's different scales at which you can study the brain. So one is sort of individual neurons, the microscopic scale. One is a typical resolution that we study with fMRI, so that the brain areas. But there is actually a scale that is intermediate, and this is of columns and lamina. And this sort of reminds me a lot of the hypercolumn idea of Hubel and Wiesel and Mountcastle, so the principal unit that sort of processes, at least in visual cortex, the information in all different ways. And these neurons are grouped in sort of columns and lamina. And so the exciting bit is that, you know, with these high resolutions, we can approach that organizational scale. One thing that I sort of like to remind myself and others of, that if you talk about brain areas, the original definition of brain areas is actually based upon differences in lamina. And so this is the work of Brockman, which most of you are familiar with, but Vogt and Vogt also studied this, not in terms of the cytoarchitecture, but in terms of the myeloarchitecture. And this is also very exciting, of course, because myelin is one of the contrasts that we can actually see in the anatomical images. One of the papers that I've found really interesting is that this paper by Velleman and Van Essen, which looks at all the different connectivities between the different visual areas, summarizes that literature. And they also published this 3-compartment model where you say, okay, ascending forwards, ascending information really terminates in the central layers, and descending or feedback layers terminates in the superficial and deeper layers. And this is really one of the interests for me for laminar fMRI. Can we actually dissociate these feed-forward and feedback connections with fMRI? And now I'm briefly going to talk about the challenges of laminar imaging. I think you've had several sessions about this. But if you look at the neural profile, you see these different layers that you can define based upon the different neurons. If you look at, say, the neurons that are actually involved in the most well-known type of neuron is the pyramidal cell, then these actually cross different layers, right? So I think that it's kind of interesting to think about the energy metabolism and how this distributes across the activity. If we look at the vascular profile, then that is also a very different profile across the cortical layers, where the bigger vessels are actually in the superficial of the surface and then penetrate the cortical surface and collect the blood back up. So one of the dominant things that we see in fMRIs is in laminar fMRI is that the signal amplitude increases across the cortical depth. And I think this is really something that, we have to think about and struggle with. So at least I do, and I'm always kind of suspicious of signal amplitude differences for that reason. So to sum up my very short introduction on laminar fMRI, I think it's a really exciting direction. And, I think this simplified three-compartment model is really, a cool tool to work with. But I think there's many sort of issues in data acquisition, data analysis, and interpretation that, of course, we're discussing here. Now, I'm going to talk particularly about population receptive fields. So first I want to say, okay, what are population receptive fields and how do we measure them? So population receptive fields is basically every neuron in visual cortex has a receptive field or process information from a small part of the visual field. This is known as the receptive field. Population receptive field just basically means that we have about 50,000 neurons per cubic millimeter. And with fMRI, we don't measure single neurons, but these populations of receptive fields. We don't only have these in the visual cortex, but we also have auditory cortex, motor cortex, or metasensory cortex. So the idea of receptive fields and population receptive fields is really a fundamental feature of the brain. So how do you measure them? We basically build a computational model of what we think the population receptive field looks like. And this is the most simple paradigm that we use over and over again. So we have a visual stimulus. It doesn't actually matter too much what it is, but this one is the one we use quite a lot. We measure the fMRI signal. This is actually an fMRI signal from V2. We measure different signals from the different parts of the cortex, and we model in the visual field what we think the population receptive field looks like. And this most simple model is a two-dimensional Gaussian, but we have different extensions and other people have different models. So if you have this model, you can actually predict when this population receptive field would respond. And so basically would respond when this bar is traveling through the population receptive field. And then you can make a prediction, taking into account the HRF. This prediction obviously is not a very good prediction for this time series. So we change the parameters of this Gaussian X, Y, and sigma to optimize the fit. So it's really a forward fitting procedure that we do. And the output of the analysis is X and Y and the sigma. We also get variance explained, how good the model fits, of course. And the X and Y parameters give us red and topic maps, which most of you will be familiar with. And PRF size will give us the size of the population receptive field. And they vary systematically across the visual cortex. So primary visual cortex, that's the smallest. V2 and V3 and V4, they become progressively larger and larger. They also increase with eccentricity, so the distance from the center of fixation. And this we can observe in every single subject within 20 minutes of scanning or so. And I think this is a nice model because to measure population receptive field properties, you need some signal, obviously. But properties, the PRF properties are independent of the PRF signal. So if you get more and more signal, your PRFs will not move more and more eccentric. You'll get better and better estimates of the size and eccentricity. So in that sense, they're independent of signal amplitude. And this is why we kind of use this approach in laminar fMRI to circumvent this signal amplitude dependence. So sum up this part. So we can model these PRF properties and they do not depend on signal amplitude. So how do PRF properties vary across cortical lamina? And this was work done by Alessio Fracasso, who is now at the University of Glasgow. And the basic idea is, well, if you look at the visual hierarchy, PRF size increases, and that is because the signal is accumulated and accumulating more across the different visual hierarchies. And so we actually thought, okay, if we look at laminar hierarchy, so when the signal comes in the central layers, and then spreads to more superficial and deeper layers, we sort of expect a similar hierarchy there. So sort of if the signal is accumulated from the central layers, the PRF sizes would be bigger. So we actually thought, okay, can we measure this? A few words on the data acquisition. So Nathalie Petriou, who is also here somewhere, has been really sort of instrumental in helping us with this. We have your custom-built surface coils. The other thing is that we simplified the stimulus. So normally we fit X, Y, and sigma, and in this paradigm, we only fit so eccentricity and size. We also modeled the stimulus to be optimal for the difference that we expect to measure. So we actually simulated the data before we started measuring it. And if we then look at the data we collected, so here's single voxel data from sort of 3 sort of deep, middle, and superficial layers. So the one thing you can immediately appreciate is that the single amplitude sort of increases towards the pile surface. Now, if you fit these data, then these are sort of the model fits that we get out. You see that the variance explained varies with the signal amplitude. You also see in this that we get measures out for PRF centers around, and we can then plot these across the cortical thickness. If you plot these across the cortical thickness, then this is the data we get out. So we see that PRF size as a distance from the gray-white boundary as this U-shaped function as we predict. Now, at the time, we had to do several simulations and control experiments to verify that this was actually the case. But what happened later is that Peter Wolfsema, who's my colleague at the Netherlands Institute of Neuroscience, actually said, well, look, we have this data also in macaques, so we can simply plot that as well. And so this is what I'm going to show you now. This is the neurophysiology results where they measured single units as a function of cortical distance. So in here, you see again the sort of the RF size in red as a function of cortical depth. And there's actually 2 things that I'm sort of very pleased about here. And taking into account that this is a very different experience, very different species, but they have a similar U-shaped relationship. And the other thing is that the units which we define our receptive field is in degrees of visual angle, so how big is it on the retina, they're actually very similar. So you see that these measurements that we make are actually similar between invasive neurophysiology. So I think this is a really nice confirmation. And it also shows this U-shaped relationship. Now, if we think about the interpretation, I talk about feed forward and feed back. So descending information arrives in the deeper and superficial layer. So you could say, okay, the PRF sizes are bigger because of the contribution of sort of feedback signals. But I think the more simple explanation is that this is simply the laminar hierarchy. So the signals come in and then are sort of pulled in a more deeper and superficial layer. So I don't think we necessarily need sort of this feedback explanation. So I think we see that the PRF size actually vary across the cortical thickness in line with this laminar hierarchical processing and in line with the sort of invasive neurophysiology. So the next thing I wanted to talk about is how do these signals vary with attention? And I think I have to a little bit explain how we think about attention. So if you take a picture with the eye, you have the fovea, which is the sharpest part of our vision. And typically what we do is we move our eyes around. But while we do this, we can still pay attention to my kids in this example. And we call this a spotlight of attention or this zoom lens of attention. And what we're here interested in, how does attention interact with population receptive fields? And I think we really need to sort of explain a little bit the modeling sort of approach. So Reynolds and Heger proposed that we can actually model attention as another receptive field. And they use it to explain things like contrast gain and response gain. So basically a multiplication of the 2, which boosts the responses. And I think this is the most well-known effect of attention. So if you look at the kids here and you pay attention to whether they're not spilling the milk, you place your attention field there, you boost the responses at that sort of location of the visual field. And this is kind of what I'm trying to illustrate here. One thing that we're actually wondering is, okay, what happens to all the receptive fields that are, outside of the intended location? Now, if you follow this model through, and if you model it with Gaussians, which is the most typical way to model the receptive fields, if you multiply 2 Gaussians that are not aligned, the resulting Gaussian will be somewhere in the middle. So if you have your attention-free receptive field and your attention field, you multiply the 2, you basically end up with a receptive field that is slightly changed its preferred position. And I think, people talk about this as the receptive fields moving. I don't think about it as moving. I think about it as changing the center of mass. And basically, you can think about it as changing the synaptic gain of the different input to the receptive fields. So what happens is a preferred position change of those receptive fields. And so if you take the spotlight of analogy again, and you say, okay, what happens to all the other receptive fields? They change their center of mass towards the focus of attention, which means that actually the cortex is actually processing the attended location more than the rest. So this is kind of similar to the magnifying glass analogy. And I think the spotlight and the magnifying glass are both, it's the same mechanism, right? They're both flip sides of the same operation. So local attention actually changes the whole visual field organization throughout the visual hierarchy. And there's several papers where we showed that this is happening. And again, this is also inspired by neurophysiology. So I think this is then a perfect way to say, look how attention actually varies across the cortical lamina, because it changes the positions. And this is, of course, something that we can measure also across cortical depth. And this was done by Barry Klein, one of my former PhD students. This is what the stimulus looks like. So we sort of basically use the same stimulus as before. We have these expanding rings. The subject fixates in the center and performs a task on these kind of tiny sort of contrast patches, which are outside the stimulus range. So it's not on the left or the right. And they detect a little contrast increment that happens sporadically every now and then. And in one scan, they attend to the right, and on the other scan, they attend to the left. So they're not changing attention. They're just, during one scan, we map out the population receptive fields while they're attending on the right. And on the time, we map out the population receptive fields when they're attending on the left. This is what the data looks like. So you see on the left where they attend to the left, you get the PRF positions or eccentricity for 2 subjects, and one time when they attend to the right. So now if you kind of look at these eccentricity maps, you can already see that there's a kind of change in the eccentricity maps without actually binning it. And if you sort of collapse all layers and all V1, for different subjects, so this is different subject and the eccentricity difference, then you can indeed see that the eccentricity estimate change depending on whether you pay attention to the left or to the right. So the next thing is then basically, okay, we collected these sub-millimeter data, and instead of collapsing it across the whole V1, we're going to collapse it across cortical depth. And this is just some data to show the anatomy, the V1 sort of ideas, and the PRF eccentricity estimates on single voxels. So if you collapse that across different subjects, so the dashed lines are different subjects, you have the eccentricity difference across cortical depth. 0 is actually the gray-white boundary. Then you see that there's a decreased slope. So the sort of the PRF position changes are the biggest in the deeper layers. So PRF attraction actually varies across cortical depth, and the PRF changes larger in the deeper layers. Now, if you think about the simplified 3-compartment model, then this is kind of an ad hoc explanation of what we think is happening. The signal comes in at the central layers, then it's spread to the most deeper and superficial layers, and the attention field operates most strongly on the deeper layers. The other thing is of interest is if you look at signal amplitude, which we can also do, is that there's actually not that much difference in signal amplitude here. And this is, you see in the contralateral hemisphere, the signal is a little bit increased, but there isn't a big difference in the signal amplitude, not as much as we see in the PRF positions, which sort of relates to this idea that you need some signal to estimate the PRF positions, but if you have more signal, the PRF positions don't systematically change with signal to noise. So to sum up this little section is that the PRF attraction is the strongest in the deeper layers in line with feedback of endogenous attention. Now, the last thing I wanted to briefly talk about is, does this model generalize? So most of the fMRI studies are done in early visual cortex or early auditory or motor somatosensory cortex. So does this sort of generalize to sort of our association cortex and the functions there? And so then I have to introduce the concept of numerosity, which we actually studied. So first, a brief explanation of what is numerosity. So numerosity is the idea that you can judge the amount of things, and you can do this automatically. Humans can do this, animals can do this, infants can do this. And basically the idea is, can you judge which place has the most cookies or which cookie has the most chocolate chips if you're so inclined? You kind of use this when you enter the classroom and say, okay, are there enough chairs for the students? The very most often used example is, okay, which apple tree the monkey has to climb in, so which apple tree has the most monkeys. The other idea is that, the apple trees may already have monkeys, so you have to also take into account the number of apples, but also the number of monkeys. And so we think this is really the sort of basics of foundations of math and economy. It's really a perceptual process, and I think this illusion sort of illustrates it as well. So the black dots and the white dots are the same amount. But if you would judge them without counting, then you would actually say, there's probably more black dots. So this is sort of to illustrate that it's still a sort of fundamental sort of automatic process, distinct from counting. And in the lab, we study these with these dots. Now to recap, so a little bit, there's lots of evidence from neural work, and fMRI that actually neurons are tuned to numerosity. So neurons prefer a certain amount, similar to preferring a visual field position. So you have receptive fields for numerosity. And if we map those with a similar procedure, we actually find these numerosity maps in the brain. And so you see the preferred numerosity very systematically across the visual cortex, and they also have a certain tuning width that goes with it. And since then, we've defined more of these maps, and we study how these maps vary and what these properties of these maps are. But the basic idea really is that we have topographic maps in these primary sensory and motor areas, but we also have them in association cortex. And if you think about why we have topographic maps, they don't only reflect this organization of the sensory motor cortices organs, but there's also computational benefits to this topographic organization. Well, I think the main point for this talk is, that these topographic principles do extend to sort of beyond primary motor and sensory cortices. And then we can actually ask, okay, are laminar properties varying in the same way in these numerosity maps as they do in, say, what we found in primary visual cortex? So these numerosity principles sort of extend to association cortexes, and the question then is, does the laminar information flow, is it actually similar in these maps as it would be in sort of the visual field maps that we've described before? And so the idea is very similar as the first experiment we talked about, where the information comes into central layers and then is sort of accumulated in the deeper and superficial layers. And this is kind of the idea that we've talked about before, that sort of across the visual hierarchy, there's a pooling and the receptive fields get bigger. Now, if you look at visual features, actually, the opposite happens. So in early visual areas, they're sensitive to the lines and shapes, and they become more and more specific as you go up the visual hierarchy. And if you treat numerosity as a visual feature, then you would actually say, okay, if it enters, it would be sort of less specific, so the tuning width would be broader. And as it gets processed more and more, the tuning width would become narrower and narrower, the response would become more specific. Because it's just a tuning to a feature and not a visual field location. So we actually predict the opposite, right? This is just a few words sort of recapping of the analysis. So we use the same surface corridor, we tie them to the head, we define cortical depth, we align the functional and anatomical data, which you can see in example here. We see here the numerosity preferences, which we reconstruct the same map across the cortical surface. This is a single time fit. So we actually get good fits for these very high resolution data. And then we have preferred numerosity versus tuning width. And so we want to do the same thing as we did in the first study, where we say, okay, how does tuning width vary across cortical depth? And so this is the results. The gray lines are individual subjects, and sort of a red line is the average of the subjects. And so you indeed see that the tuning width as a function of cortical depth has this inverse U shape. And this actually suggests that, okay, the model that we have in V1 may apply to at least this numerosity map in parietal cortex, suggesting that perhaps this is indeed sort of a model that may transfer to other areas beyond primary visual and motor cortices. I think this is the sort of the summary of the last study. So the topographic principles, they extend, and which allow us to use actually the same sort of paradigms that we have used in primary visual cortex, in this case for the numerosity maps, and we see a very similar result there, supporting this canonical laminar organization. And last, I should thank all the people who have contributed to this, people at Spinoza, and at and my lab, and in particular Alessio, Jelle, and Barry, who did all the work. And thank you for your attention.